One major cause of blindness in the world today is a category of diseases known as glaucoma. Glaucoma is characterized by a raised inner eye pressure, which puts a strain on the optic nerve. The optic nerve is located at the back of the eye. The cornea is on the front of the eyeball and covers the iris, pupil, and the anterior chamber. The iris's primary function is to dilate, which regulates the amount of light that passes through the pupil and onto the lens. The pupil is the gap at the center of the iris. The space in front of the iris and behind the cornea is known as the anterior chamber, which is full of a clear liquid called the aqueous humor. In a healthy eye, the amount of fluid in the anterior chamber is regulated by a spongy tissue known as the trabecular meshwork. The anterior chamber should have just enough fluid in it to give the cornea a slight bulge. The trabecular meshwork lets the fluid flow through and passes it on to a microscopic tube. This tube is called the Schlemm's canal, which eventually flows out to the bloodstream. If the trabecular meshwork is working properly, it should let through most of the aqueous humor, but keep enough inside the anterior chamber to leave it inflated. In the case of open-angle glaucoma, the trabecular meshwork doesn't let through enough of the aqueous humor. The aqueous humor that is not let through by the trabecular meshwork is left to build up inside the anterior chamber. The buildup of extra fluid overinflates the anterior chamber, which effectively raises the inner eye pressure. Raised inner eye pressure, otherwise known as high intraocular pressure, puts a strain on the optic nerve. This strain damages the optic nerve, which is what eventually leads to the loss of vision. Usually, patients lose their peripheral vision first. The so-called tunnel vision gets worse and worse over time, and almost always leads to blindness if left untreated. Although it cannot be cured, the symptoms of glaucoma can be halted or at least drastically slowed. Unfortunately, there is no known treatment strategy that restores vision after it has been lost to glaucoma. Ideally, the glaucoma is caught before too much loss of vision has occurred. If it is caught, then the primary method of care is simple medications, usually in the form of eye drops. The medications are designed to lower intraocular pressure, usually by increasing the flow of fluid through the trabecular meshwork, but occasionally decreasing the amount of aqueous humor produced. Often the medication becomes ineffective and a surgery is necessary. The surgeries are also designed to lower inner eye pressure. The two types of surgery most commonly used are conventional surgery and a laser tuberculoplasty. The laser surgery is the one used by default, but only works if there is a clear cornea. The laser makes burns in the net meshwork and stretches it out, usually successfully lowering the intraocular pressure. Depending on how advanced the glaucoma is, the surgery keeps the inner eye pressure lowered for varying lengths of time, but is almost always not permanent. Conventional surgery, otherwise known as a trabeculectomy, is only done once medication and laser surgery has failed to work. This surgery works by physically cutting a gap in the tissue where the fluid can drain through. This lowers inner eye pressure. There are many different types of glaucoma, the most common being primary open-angle glaucoma. This is almost always developed by people over the age of 40, but when it is developed in childhood or young adult years, it is called juvenile open-angle glaucoma. Even more specifically, there is a rare condition when glaucoma is developed before the age of 5, and it is called primary congenital glaucoma. For this presentation, primary congenital glaucoma will be referred to as PCG. PCG is an autosomal recessive genetic disease. The exact cause for PCG is unknown, but research has shown that 20 to 40% of all PCG patients have a genetic mutation in the CYP1B1 gene. This gene codes for a protein that is commonly found in the trabecular meshwork and other parts of the eye, but the reason it causes the glaucoma is still unknown. One possible treatment strategy for glaucoma is the procedure of gene therapy. Gene therapy works in theory, but is a highly debated subject. It has been mostly successful in labs and has moved on to do thousands of clinical trials, but it can still be dangerous. In primary congenital glaucoma, there are two major physical barriers to doing gene therapy. First, there is the strategy of actually getting the new gene into the cell. Second, There are the possible reactions by the human body to the new gene. In order to successfully perform gene therapy, you need a way to insert the DNA into the target cell. Vehicles that are used to insert the DNA to the cells are called vectors, which are divided into the two major categories, viral and non-viral. Both types of vectors have been tested on ocular gene therapy with mixed success. Some examples of viral vectors that have been tested include adeno-associated virus, adenovirus, lentivirus, and retrovirus. A recombinant adeno-associated virus has moved on to clinical trials due to success in animal tests. In general, viral vectors have a higher success rate in getting the gene to the new cell than non-viral vectors, and they do so with more efficiency. Unfortunately, viral vectors can become toxic to the cells and often create an inflammatory or immune response. For these reasons, research has shifted to non-viral vectors for ocular gene therapy. Non-viral vectors are generally cheaper to use and are less toxic to cells. They also cause less immune response and less inflammatory responses than viral vectors in the case of ocular administration. In non-viral gene therapy, the DNA doesn't have a virus to carry it to the cell, but it still needs some sort of chemical escort to get it there. Some examples of said chemical escort include lipoplexes and polyplexes, which condense the DNA and carries it to the target cell. Polyplexes and lipoplexes are complexes of polymers, or liposomes, respectively, that are held together using electrostatic interaction. But the ethics of gene therapy also prevent its widespread use. Gene therapy can be dangerous for several reasons. For one, there is a chance of inducing a tumor, and also a chance of immune response. Thirdly, of course, there is always the chance that the virus in a viral vector recovers the ability to cause a disease. In fact, 2 people have died in clinical testing of gene therapy. Unfortunately, this type of gene therapy doesn't last for very long because the unaffected cells will divide. The therapeutic targeted cells must be long-lived and stable. Problems with integrating therapeutic DNA into the genome and the rapidly dividing nature of many cells prevent gene therapy from achieving long-term benefits. Patients usually must undergo multiple rounds of gene therapy. It is worth clarifying that gene therapy in the eye would not be passed down to future generations. The only situation where the children would also express the gene that was inserted to the parent's cell would be if the gene therapy was performed on the egg cells or sperm cells, which is known as germline gene therapy. Doing this type of gene therapy would be extremely dangerous because we don't necessarily always know all the different outcomes of gene therapy. We're not sure if changing one gene could affect other genes too. The Human Genome Project showed us that each gene codes for much more than just one protein. So if it's one protein that is malfunctioning, it's possible that changing the gene would affect much more than just that protein. For these reasons, many people argue that we shouldn't do any gene therapy, because we don't exactly know what the outcomes are going to be. To summarize, glaucoma disease is a disease that degrades the eyesight of patients. It is characterized by a raised inner eye pressure, which puts a stress on the optic nerve and results in vision loss. Glaucoma is not curable, but can be prevented and suppressed through simple medication and eye drops. If these methods become ineffective, surgeries can slow or even halt the onset of glaucoma for a long period of time. Gene therapy is a promising method of stopping glaucoma, but so far there are too many drawbacks. Current methods have too much risk for widespread public usage.